Imagine a scenario for a second. You're looking at a pregnant mother, right?
Yeah.
And she catches a severe, but honestly incredibly common viral infection.
Like a bad flu or something like that.
Yeah, exactly. A bad flu. And her immune system does its job. It fights it off. She recovers. And the rest of the pregnancy just continues totally normally.
Right.
But then fast forward, I don't know, 15 or 20 years. And her child is suddenly diagnosed with schizophrenia.
Which is a devastating diagnosis.
It really is. And for decades, we've looked at those two events and wondered, is this just a tragic, random coincidence? Or is there a direct physical biological wire connecting that distant maternal immune response in the womb to the teenager's brain architecture today?
Yeah, and it's really one of the most profound mysteries in neurobiology. I mean, the medical community has observed this statistical correlation for a very long time.
Right.
We've known that severe maternal infections significantly increase the risk of neurodevelopmental disorders. So things like autism spectrum disorder, bipolar disorder, schizophrenia.
But the actual mechanism.
Exactly. That has always been a complete black box. Like, we could see the starting line and we could see the finish line, but the entire race in between was just hidden from us.
Well, today's deep dive is all about taking a crowbar to that black box. We are unpacking a genuinely groundbreaking 2026 article from the journal Neuron by a research team led by Bizzotto and colleagues.
A fantastic paper.
It really is. And our mission today is to trace this exact cellular domino effect. We're going to go molecule by molecule, starting from the mother's immune system, pushing across the placenta, and going right into the intricate wiring of the fetal brain.
And we really have to appreciate the level of resolution we have now, because historically, science could only look at, broad associations, like tracking hospital records.
Yeah, epidemiological stuff.
Right. Yeah. But what this team accomplished is staggering. We are looking at the specific genetic instruction manuals of individual microscopic cells at the exact moment the trauma occurs.
Okay, let's unpack this. If we're tracing dominoes, we have to start at the very first one, which is the mother's immune response. Now, in this study, the researchers didn't just cough on a mouse and hope it got sick.
Oh, no. They needed absolute precision. So they used a highly controlled experimental model called maternal immune activation, or MIA.
Okay.
To trigger this, they took wild-type pregnant mice at exactly gestational day 15, which in human terms corresponds roughly to the late first or early second trimester.
Which is a hypercritical window for brain development, right?
Exactly. It's when the foundation is really being poured. So they injected these mothers with a substance called poly-IC.
And just to clarify what poly-IC is for you listening, because this totally fascinated me.
Yeah.
I like to think of it as a biochemical fire drill.
That's a great way to put it.
Because it's a synthetic, double-stranded RNA molecule. So there's no actual replicating virus in the mother's body, no pathogen that's going to actually eat her cells.
Yeah.
But her immune system detects this specific shape of RNA and gets completely tricked into thinking it's under a massive viral attack.
It's the perfect decoy. And, the body reacts exactly as it would to a real threat. The biological fallout is immediate and explosive.
How immediate?
Within just 6 hours of that injection, the pregnant mother's blood plasma is absolutely flooded with interleukin-6, which is a classic inflammatory marker.
Okay.
But the real star of the show here, the critical domino, if you will, is a massive elevation of type I interferon, specifically interferon beta.
Type I interferon. Okay, whenever I read about interferons, I always picture the body's primary emergency broadcast system.
Yes.
It's like the signal a cell sends out when it's breached, basically screaming at all the neighboring cells to batten down the hatches.
That's a highly accurate way to look at it. When that broadcast goes out, the surrounding cells immediately pause their normal operations, and they just start pumping out specialized defensive proteins. Yeah, the researchers looked at the mother's brain, specifically the hippocampus, and they saw these massive spikes in what we call interferon-stimulated genes, or ISGs.
And the paper lists a whole alphabet soup of these genes, right? Like MX1, ERF7, IFN-3, IFN-M3, ISG15, STAT1, CH25, but what are they actually doing?
Think of them as the cellular riot police. Some of these genes produce proteins that physically block viruses from entering the cell membrane.
Okay.
Others act like biological shredders. They hunt down foreign RNA and literally chop it up. The mother's body is flawlessly executing millions of years of evolutionary defense programming.
And here is where I really have to push back, or at least point out the tragic irony of this whole thing.
Go ahead.
The mother's immune system is doing exactly what it's supposed to do. It's sounding the alarm, deploying the riot police, keeping the host alive. How does a perfectly natural protective defense mechanism accidentally become a weapon against the baby's developing brain?
That is the million-dollar question. And it really forces us to rethink how we view illness. Because we tend to think of the pathogen, the virus or bacteria, as the sole destructive force.
Right.
But fetal brain development requires an incredibly delicate, quiet, and precisely timed chemical environment. A symphony of hormones and growth factors.
So the noise is the problem.
Exactly. When the mother's immune storm rolls in, the sheer volume of inflammatory chemicals drowns out the symphony. The defense itself is the disruption.
Okay, let's pause here and do a quick bullet summary for you listening at home because we are covering a lot of ground.
Good idea.
So, bullet 1, poly-IC acts as a viral mimic. Bullet 2, it triggers a massive type I interferon immune response in the pregnant mother. And bullet 3, the mother's brain activates a cascade of specific antiviral genes.
Perfect summary.
So here's a question for the audience to chew on. Have you ever considered that an organism's response to an illness might be more structurally impactful than the illness itself?
It's a wild thought.
It really is.
Yeah.
Okay, moving to the next section. Imagine you're designing a security system for a developing brain. Your biggest nightmare isn't just a virus slipping in, it's the mother's own security system panicking and breaking down your doors.
Right.
Which leads to the obvious next question. The mother is having this immune storm in her own bloodstream. How does that biological panic physically cross over into the fetal environment?
To figure that out, the researchers use this fascinating piece of technology called FACS. That's FACS, which stands for fluorescence-activated cell sorting.
Oh man, I love the magic of this machine.
Yeah.
Can you explain how it works for the listeners?
Sure. Imagine taking a tissue sample and you break it down into a liquid soup of millions of individual cells.
Okay.
Then you shoot that liquid in a single file line through a laser beam. But beforehand, you've tagged specific proteins on the cells with glowing markers.
Like little neon signs.
Exactly. So the FACS machine looks at each individual cell as it flashes by the laser, identifies what glowing uniform it's wearing, and physically sorts it into a different tube. And it can do this to thousands of cells a second.
So they are literally sorting the cells one by one to find out who heard the mother's alarm.
Yes, and they looked specifically at the embryonic yolk sac, which at this stage of development is acting as a crucial barrier and life support system.
Right.
They isolated the macrophages there. Macrophages are basically the immune system's border patrol guards. And they discovered that these specific fetal border guards, the ones labeled CX3CR1 plus F480 low and high, were completely lit up.
They were glowing.
Yeah, they were expressing massive levels of a membrane protein called IFN-I, which, if you remember, is one of those interferon distress signals.
So the fetal boundary cells are physically absorbing the mother's panic. They heard the broadcast. Yep.
They absorbed it, and then they passed it on. And the timeline here is what truly shocked the neurobiology community. The researchers detected an interferon burst inside the actual embryonic brain just 3 hours after the mother received the poly-IC injection.
Wait, 3 hours?
3 hours.
From a shot in the mother's abdomen to a genetic alteration in the fetal brain. That is terrifyingly fast.
It's lightning fast. The embryonic brain immediately starts transcribing those same alarm genes, ERF-F7, IFN-I. But here is the most elegant, subtle biological detail.
Hit me.
The receptor on the fetal brain cells that actually catches this signal, a receptor called IFN-I, it doesn't increase in number.
Oh, really?
No, the physical hardware stays exactly the same. It is simply the activation of the pathway that echoes across the barrier. The hardware is normal, but the software is suddenly forced to run an emergency protocol it wasn't scheduled for.
Okay, time for our second mini recap.
Go for it.
Bullet 1. The maternal interferon alarm physically reaches the placenta and yolk sac. Bullet 2. Yolk sac macrophages absorb the signal via IFN-I. And bullet 3. The embryonic brain registers the immune panic just 3 hours after the mother's exposure.
Spot on.
And a question for you listening. If you were engineering a protective barrier for a developing brain, how would you filter out the mother's biological panic signals without ignoring genuine threats?
It's basically the ultimate biological engineering problem.
Totally. Okay, so the panic has breached the barrier. The fetus's brain is now running this emergency software. But what happens next? Because schizophrenia isn't something you see in a newborn.
No, it typically emerges much later.
Right, it shows up in adolescence or adulthood. So let's fast forward to when the baby mouse is born and his brain is actively trying to wire itself up.
This is where we look at what we call the postnatal chaos. In the study, they focused on juvenile mice at postnatal days 8 and 18, so we'll call them P8 and P18. This window is critical because this is when the brain is furiously building and then purposefully destroying neural connections.
Right. They measured the density of excitatory synapses using PSD95 density. These are the connections that allow neurons to fire and stimulate each other.
Exactly.
And the results were wild. At P8, the offspring of the immune-activated mother showed this chaotic, abnormal overgrowth of excitatory synapses.
Like a jungle.
Yeah. But then just 10 days later, at P18, there is a massive damaging loss of those exact same synapses. It's like a crew building a skyscraper twice as fast as they should, only to take bulldozers to the whole thing a week later.
It's completely erratic.
So what is driving that erratic behavior?
The culprit is a tiny receptor on the surface of the microglia. Now, microglia are the brain's resident immune cells, but they also act as the brain's janitors and architects.
Multitaskers.
Very much so. And the receptor in question is called TREM2. That's TREM2.
TREM2. Okay, we need a way to visualize this because TREM2 is the absolute linchpin of this entire deep dive. So if the microglia are the construction crew, is TREM2 like a traffic cop?
Well, a traffic cop implies it's just directing flow. Think of TREM2 more as the on-site construction foreman.
Oh, okay.
When TREM2 is present and functioning properly, it reads the blueprints. It directs the microglial crew on exactly which neural connections to reinforce and which to demolish, which is a vital, healthy process we call synaptic pruning.
The foreman. I really like that image. So what happens to the foreman when that prenatal immune storm hits?
Under normal conditions, TREM2 is firmly anchored to the outer membrane of the microglia cell. He's on the job site wearing his hard hat.
Right.
But the researchers found that the maternal immune burst severely downregulates this membrane-bound TREM2, essentially rips it off the surface. And at the same time, they found incredibly high levels of a broken, soluble version of TREM2-STREM2 floating away in the offspring's blood serum.
Oh, wow. Okay. Keeping with our visual, the maternal storm sweeps through the construction site and knocks the foreman's hard hat off.
Yes.
The hard hat, which is the receptor, washes away down a river into the blood. So now you have a crew of microglial bulldozers with no foreman. They go totally rogue.
Exactly.
They overbuild synapses at P8 because nobody's telling them to stop. And then at P18, they just start indiscriminately destroying critical connections.
That perfectly captures the destruction. And it presents a really fascinating biological paradox.
What's that?
Well, the microglia's physical ability to eat or prune synapses, their phagocytic function, is clearly hyperactive and broken. But later in the study, when researchers managed to artificially block the maternal interferon, it restored the TREM2 foreman to the job site. The synapse density went back to normal, but it did not fix the microglia's underlying eating behavior.
Wait, let me wrap my head around that. They fixed the building, but the crew was still acting crazy.
Exactly. It proved that the loss of synapses and the aggressive eating behavior are actually decoupled. They are independent pathways that were just triggered by the same initial prenatal alarm.
That is mind-blowing.
And importantly, because that broken hard hat washes into the blood, that soluble TREM2 actually serves as a measurable biomarker. You can test the blood to see that deep brain dysfunction is occurring.
Let's do a quick recap 3 for the listeners. Bullet 1. Prenatal immune alarms cause postnatal loss of functional TREM2. Bullet 2. Without TREM2, microglia act chaotically, overbuilding then destroying synapses. And bullet 3. Soluble TREM2 in the blood serves as a biomarker for this dysfunction.
Excellent.
And an audience question. If TREM2 is the foreman of your brain's architecture, what happens to the structural integrity of a city when the boss goes missing during peak development?
Chaos. Total chaos.
Yeah. But, science is all about rigorous proof. How do they prove beyond a shadow of a doubt that missing this specific foreman, TREM2, is the sole reason the skyscraper collapses?
They use the gold standard of genetic testing, the knockout experiment.
Love a good knockout experiment.
They engineered a line of TREM2 mice, meaning they completely lacked the TREM2 gene from birth. No foreman at all, ever.
Okay.
They then compared these knockout mice against genetically normal mice, subjecting both groups' mothers to the same poly-IC viral mimic.
And this is where they use that single nucleus RNA sequencing, right?
Mm-hmm.
I want to pause on this because it sounds incredibly dense, but the magic of this tech is wild. They aren't just looking at a chunk of brain tissue.
No.
They are freezing a moment in time and reading the exact biological instruction manual being actively used inside a single microscopic cell nucleus.
Precisely. They zoomed in on the P18 hippocampi, focusing exclusively on glutamatergic neurons.
Which are the excitatory neurons that dictate brain activity.
Yes.
Yes.
And in the genetically normal mice whose mothers experienced the infection, this sequencing revealed a massive spike in gene pathways tied directly to human schizophrenia, autism, and bipolar disorder. The damage was written right into the RNA.
But here is the twist that absolutely broke my brain when I read it. In the knockout mice, the ones completely missing the TREM2 foreman, the maternal infection did not cause those massive transcriptomic shifts. Let that sink in for a second.
It is deeply counterintuitive. The logical assumption is that if you remove an important piece of brain architecture, the damage from an immune storm would be much worse.
Right.
But instead, TREM2 actually acts as a biological gate. The environmental damage from the maternal infection absolutely requires the presence of TREM2 to fully manifest its toxicity in the neurons.
So lacking a crucially important brain gene actually protected them from this specific environmental trauma. Did this translate to how the mice actually behaved?
It translated perfectly. When the normal mice grew up, they showed extreme, debilitating anxiety. They spent significantly more time hiding in dark chambers during behavioral tests.
Okay.
But more importantly, they exhibited a very specific trait that mimics human schizophrenia, a reduced prepulse inhibition of the startle reflex. We call it PPI.
Let's explain PPI quickly, because it's a brilliant way to measure how a brain processes reality. If you hear a small sound, like a pencil tapping on a desk, right before a loud sound, like a heavy book dropping,
Right.
a healthy brain uses that small sound to prepare itself. You don't jump as high at the loud noise. Your brain filters out the sensory chaos.
Exactly. But schizophrenic patients often lack this filtering ability. Every loud noise is a completely new, terrifying surprise, regardless of the warning.
Wow.
The normal immune-activated mice showed this exact same sensory filtering failure. But the TREM2 knockout mice, they did not develop these behavioral deficits.
Again, the paradox of vulnerability.
It's worse than just a paradox. It's a fundamental misunderstanding of pathology. Without TREM2, these mice are actually primed for an exaggerated initial immune response. They totally lose a vital microglial state called MIC.1.
Which is the normal, healthy, interferon-responsive resting state.
Right. But because they lack the TREM2 gatekeeper, the downstream cascade that causes the schizophrenia-like behavioral damage never triggers. Missing a piece of the puzzle prevented the entire tragic picture from forming.
Okay, mini recap for bullet 1. Single nucleus RNA sequencing proves maternal infection alters neuronal DNA reading. Bullet 2. Normal offspring develop anxiety and schizophrenia-like startle reflexes. Bullet 3. Mice lacking TREM2 are paradoxically shielded from these adult behavioral deficits.
That's the core of it.
And a question for you at home. Isn't it a fascinating paradox that missing a core functional gene might actually act as a shield against a highly specific environmental trigger?
It really makes you question what we call a defect.
Absolutely. But as fascinating as this is, I keep thinking everything we've discussed so far relies on mice, poly-IC, knockouts, P18 hippocampi. The ultimate test of this mechanism is whether this exact cellular domino effect happens in human beings.
Yes.
Does it translate to us?
The researchers knew they had to cross that bridge, and they brought in an incredibly validating and frankly chilling piece of evidence.
What was it?
They examined postmortem brain tissues from 35 human patients who had been diagnosed with schizophrenia during their lives. They looked specifically at a region called the superior temporal gyrus.
35 human brains. What did they find when they looked at the biological instruction manuals of those human cells?
They found profoundly suppressed type I interferon responsive genes. We're talking about the exact same genes we discussed earlier, STAT2, I5TM3, and IRF7.
Oh my God.
It perfectly, almost hauntingly mirrors the destroyed MIC-POT1 microglial state that they observed in the juvenile mice.
That is the smoking gun. Think about the gravity of that for a second. You are looking at the physical brain tissue of a 35 or 40-year-old human adult, and you are tracing a biological scar all the way back to a viral alarm that went off while they were still in the womb.
It connects a decades-old maternal infection directly to adult psychiatric architecture. But, you know, the beauty of the Bizzato paper isn't just that they mapped the damage. They used this map to design a rescue mission.
If you know exactly how the dominoes fall, you can put your hand in and stop them. So how did they intervene?
The intervention was brilliantly targeted. They went back to the pregnant mice. Exactly 24 hours before they injected the poly-IC viral mimic, they administered a monoclonal antibody directly to the mother.
Oh.
This antibody was specifically designed to block the type I interferon receptor. In the literature, it's called alpha IFN-IR.
So they essentially put biological earmuffs on the mother's immune receptors before the fire alarm even went off.
That's exactly what it did. This block neutralized the maternal interferon burst. The alarm rang, the mother's body registered the fake virus, but the specific biological pathway couldn't amplify the panic signal.
And the results in the babies.
Miraculous. The antibody treatment completely restored normal wiring in the offspring. They proved this using patch clamp recordings on the neurons.
Which physically measures the electrical currents in a single cell, right?
Right, and it showed that the excitatory to inhibitory ratio, the crucial EI balance, was completely normal. It rescued the membrane levels of the TREM2 foreman.
So the foreman got his hard hat back.
He got his hard hat back, the crew went back to work normally, and furthermore, the intervention normalized the CDK8 and pSTAT1 protein axis.
What does that axis do?
That axis is the specific molecular pathway that regulates the brain's tonic interferon response. You can think of a tonic response as the low-level hum of the electrical grid, rather than the blaring fire alarm. The whole system was brought back to peace.
Okay, final recap before we wrap up. Bullet 1. Human schizophrenic brains show the exact same suppressed microglial gene states as the infected mice. Bullet 2. Blocking the mother's IFN-I receptors prior to the immune spike completely rescues the baby's brain wiring. Bullet 3. The CDK8 pSTAT1 pathway is identified as the therapeutic target.
Perfect.
And our last audience question. If medical science could safely monitor and tweak your immune response levels during pregnancy, would you want to know if it meant preventing a neurological disorder in your child?
I think most people would say yes.
Right. Here's where the clinical implications of this just blow my mind. Currently, if someone is diagnosed with a neurodevelopmental disorder like schizophrenia or severe autism, we try to treat them in their late teens or adulthood.
After the damage is done.
Exactly. We are prescribing medications long after the skyscraper has been structurally wired with missing foremen. But this study suggests a paradigm shift. Imagine a future where part of standard prenatal care involves monitoring specific maternal immune markers, like type 1 interferon, in real time.
It's totally possible.
If a pregnant mother catches a bad flu, we don't just tell her to rest and drink fluids. We use targeted, safe antibodies to protect the baby's brain wiring before the damage ever occurs.
It shifts our entire medical philosophy from symptom management in adulthood to molecular prevention in utero. It is an ethical and medical frontier that we are rapidly approaching.
Let's pull all the way back and look at the macroscopic view of what we've unpacked today. A pregnant mother experiences a viral mimic. It triggers a type 1 interferon immune storm. That chemical panic breaches the placental border guards within hours, reaching the fetal brain.
Yep.
Weeks later, that early alarm strips the hard hat off the TREM2 construction foreman in the baby's brain. Without their foreman, the cellular construction crew goes rogue, leaving behind chaotic synaptic wiring and sensory filtering failures that perfectly mirror human schizophrenia, and we now know we have the potential to intercept that signal.
It really is a triumph of modern science. But, it leaves us with something very deep to consider. We now see with undeniable clarity that early immune memories strictly dictate our physical brain architecture, which means every single one of us is walking around with a brain built by our mother's immune history. It raises a question I think everyone listening should explore on their own. What other microscopic, forgotten environmental alarms are our brains quietly remembering right now? Are our very personalities, our anxieties, our unique cognitive quirks partly shaped by the invisible viruses our mothers successfully fought off before we ever took our first breath?
That is, honestly, it's a little haunting. The idea that we are the living echoes of battles we weren't even awake for. But remember that murky black box of a diagnostic landscape we started with? Thanks to studies like this, the picture is finally coming into focus, cell by cell. Thank you for joining us on this incredible deep dive into the frontier of neuroimmunity. We'll see you next time.